Introduction, Professor. In today's video, we will explore rickettsial infections caused by a group of bacteria called Rickettsia. These diseases are transmitted by arthropod vectors and can range from mild to severe. We will delve into the molecular mechanisms of pathogenesis and discuss potential treatments. Let's get started. Overall, understanding the complex molecular mechanisms employed by rickettsial bacteria to invade and survive within host cells is crucial for developing effective treatments and preventive strategies for rickettsial infections. Professor, macrophages play a critical role in the immune response against rickettsial infections. Upon infection, Rickettsial stimulate the activation of macrophages, which are vital in clearing pathogens through phagocytosis and the secretion of various pro-inflammatory cytokines. However, Rickettsial employ strategies to evade macrophage-mediated killing, such as inhibiting phagolysosomal fusion and modulating host cell apoptosis pathways. Understanding the intricate dynamics between Rickettsial and macrophages is fundamentally important for designing therapeutic strategies that promote an effective macrophage response and limit bacterial survival. Professor, T-cell immunity is another crucial aspect of the host defense against rickettsial infections. Rickettsia-specific T lymphocytes are crucial in controlling bacterial replication and preventing disease progression. These T cells recognize rickettsial antigens presented on infected cells and activate effector mechanisms to kill bacteria. The delicate balance between protective T-cell responses and immune-mediated tissue damage is essential in rickettsial infections. Understanding the antigen presentation pathways and effector functions of T cells during rickettsial infections provides insights into developing improved vaccines that induce strong and long-lasting immunity. Professor, host factors, including genetic variations, play a significant role in determining an individual's susceptibility to rickettsial infections. Certain polymorphisms in specific genes involved in immune responses or cellular processes may increase or decrease the likelihood of infection and disease severity. Identifying these genetic factors is crucial in predicting individual susceptibility, evaluating disease outcomes, and implementing personalized treatment approaches. Professor, antibiotic therapy is the cornerstone of rickettsial infection treatment. Understanding the pharmacokinetic and pharmacodynamic properties of antibiotics is crucial for optimizing drug efficacy and minimizing the emergence of antimicrobial resistance. Factors such as antibiotic penetration into host cells, intracellular drug targets, and optimal dosing regimens need to be considered for successful treatment outcomes. Moreover, the development of antibiotic combinations and alternative treatment options is essential in cases where drug resistance or treatment failures occur. Professor, accurate and timely diagnosis of rickettsial infections is critical for appropriate patient management and public health interventions. Molecular diagnostic techniques, such as polymerase chain reaction, PCR, and sequencing, have revolutionized the identification of rickettsial species and strains. These methods enable rapid detection, facilitate epidemiological studies, and aid in identifying novel or emerging rickettsial pathogens. Professor, rickettsial infections often trigger immune-mediated pathology, contributing to disease severity and complications. The immune response, particularly the production of pro-inflammatory cytokines, contributes to tissue damage observed in organs affected by rickettsial infections. Understanding the immunopathological mechanisms involved is crucial for developing strategies to attenuate excessive inflammation while preserving host defense mechanisms. Professor, genomic epidemiology allows for the tracking of rickettsial outbreaks and the characterization of strain diversity. By analyzing whole genome sequences, researchers can identify outbreak sources, assess transmission patterns, and uncover relationships between different rickettsial strains. This information is valuable in guiding public health interventions, including vector control measures and implementation of preventive strategies. Professor, co-infections involving rickettsial pathogens and other infectious agents are increasingly recognized. These co-infections can result in complex interactions between different pathogens and the host, influencing disease severity and treatment outcomes. Understanding the immune interactions during co-infections is crucial for managing patients effectively and preventing potential complications. Professor, vector competence refers to the ability of arthropod vectors to acquire, maintain, and transmit rickettsial. Factors influencing vector competence include vector physiology, feeding behavior, and the presence of symbiotic microorganisms within the vector. Investigating vector competence is essential for understanding the transmission dynamics of rickettsial pathogens and developing effective vector control strategies. Professor, the emergence of antibiotic resistance in rickettsial pathogens underscores the need for the development of novel antimicrobial agents. Researchers are exploring various approaches, such as identifying new drug targets, repurposing existing drugs, and developing combination therapies to overcome resistance and improve treatment outcomes. These advancements are vital to stay ahead of the evolving challenges posed by rickettsial infections. Professor, specific populations, including pregnant women and immunocompromised individuals, present unique challenges in the management of rickettsial infections. The physiological changes associated with pregnancy and the compromised immune system of certain individuals require tailored approaches to diagnosis, treatment, and prevention of rickettsial diseases. Understanding the nuances of rickettsial infections in special populations is crucial for optimizing patient care. Professor, in summary, we have discussed various aspects of rickettsial infections, including their pathogenesis, treatment, and prevention. While these diseases present challenges, ongoing research and collaboration shows promise in improving our ability to manage them. Thank you for your attention and continued interest in this field.